My name is Daniel Depledge. I'm a group leader here at the Hanover Medical School. My specialty is systems virology and I'm head of the research group focusing on that. In general, we're very interested in a group of viruses called herpes viruses. Most of us have these viruses in us, they infect us when we're young, and they establish a state of hibernation within us and they stay with us for life. However, if you're immunocompromised or if you become immunocompromised, these viruses can reactivate and they can cause very, very serious disease or even lead to death. So we are very interested in understanding what are the cellular mechanisms that help to regulate whether or not this virus can stay in hibernation or come out of it, and try to find ways of treating this with different drugs. Within the research cluster, I'm focusing on varicella zoster virus and human cytomegalovirus. So these are very different types of virus. They both establish latency in very different parts of the body. But we think some of the mechanisms that control their ability to stay latent are very similar. So we're trying to identify these mechanisms that regulate these viruses in common and then evaluate those as drug targets that can be used for developing therapies in the future. Trying to understand how they actually manipulate the cellular environment to effectively cloak themselves, to make themselves invisible to detection, yet able to also react to external stimuli such as stress or a change in immune status that then allows them to reactivate. We don't know all the pathways that are involved in this, but we know some of them, and these are the ones which initially we're focusing on. The long-term objective is to find drugs or treatments which can either prevent the virus from ever reactivating, coming out of hibernation, or even better, to clear the virus from the body completely. Principally because these viruses have been evolving with us for tens of thousands, hundreds of thousands of years. They are exquisitely poised to kind of interact with our immune system. They know how to evade sensing, and they're very difficult to do experiments on that, experiments in the lab that we can then translate into people. We can't do experiments in people, so we try to develop different types of models for developing our drugs, and then we can take them into people at a later stage. So I guess what really fascinates me is the fact that, over the last 50 to 100 years, we have accumulated huge amounts of knowledge about the human body and about these viruses. And yet it's a drop in the ocean compared to what we need to know about them to really understand how we interact with them, both as individuals and also as a kind of a wider human population. I think whenever we start any endeavor in human society, we don't know what we're doing. Every little thing is taking a step into the light, is trying to shine more light in this big cave or whatever that we're exploring. And that's what's so great about this research. And the way technology is developing, the way scientific collaboration is developing, we can really expand our frontiers very quickly now. There is a great opportunity, as we've seen with the coronavirus epidemic and the rapid response in terms of developing drugs and vaccines and treatments to preserve lives. And taking that and trying to integrate that into the wider scientific community in terms of how we investigate other viruses, that could really change things very quickly. Science is best when there is a diversity of input. So being able to collaborate with world-renowned experts in all these different parts of viral biology and human response to viral biology, that's what makes being part of a cluster like RESIST so powerful. I can't know everything there is about these viruses, so it's hugely important that I can surround myself with people who know all these different parts, and together we can put this puzzle together and really understand how it works. One of the key things that brought me here was the quality of the scientists that work here. There's a great potential to collaborate with many different people. There is a huge diversity of knowledge which allows you to apply different perspectives to a problem. And most importantly, working with these people who all hold different parts of a puzzle allows us to put them together and to really understand how viruses and humans are interacting.